Good morning. We're presenting our AP Biology capstone on two of the most common neurodegenerative disease categories, Dementias and Parkinsonisms.
Over the next several minutes, we'll show you what makes these two disease groups different at the molecular level, why that difference matters clinically, and how the AP Biology curriculum we studied this year directly explains both.
We started this project with a single question. Both Alzheimer's and Parkinson's destroy neurons. They both also progress over years and have no cure. But one strips away somebody's memory, while the other strips away their ability to walk. The clinical pictures are also nothing alike, so our entire investigation really came down to explaining why and how.
This table summarizes our presentation. Each row is one variable. The left column shows what happens in dementias, the right column shows us what happens in Parkinsonisms, and the one to the very right shows similarities in both. Symptoms diverge first. Dementias present as memory loss and cognitive decline. Parkinsonism presents as tremor, muscle rigidity, and slowed movement. The diseases hit different brain regions. The hippocampus and cortex for dementias.
The substantia nigra for Parkinsonisms.
They involve different misfolded proteins, amyloid beta and tau for dementias, alpha-synuclein for Parkinsonisms, and they disrupt different neurotransmitters, acetylcholine versus dopamine.
Before we talk about the biology, let's talk about the scale. In the United States today, 7.2 million Americans over the age of 65 are living with Alzheimer's, and 1.1 million are living with Parkinson's. Dementia care alone costs $384 billion per year, and these figures are projected to roughly double by 2050 as the general population ages. These numbers are not just abstract, as most people in this room will know somebody affected by one of these diseases, or already do know somebody affected by one of these diseases.
Here's our first major AP biology connection. Unit 1 taught us that a protein's function is determined by its three-dimensional shape. Both diseases begin when that shape goes wrong. Step 1. A protein begins as an amino acid sequence that folds into its correct shape. Step 2. Through age, oxidative stress, or genetic mutation, the protein folds into an aberrant structure. Step 3. Misfolded copies clump together with other misfolded copies, forming aggregates. Step 4. These aggregates overwhelm the cell's protein recycling machinery. The neuron loses function and dies. Critically, the misfolded proteins can induce neighboring healthy proteins to also misfold. This is why neurodegenerative diseases spread through brain tissue over years.
Both diseases share the same mechanism, then why are they different? The answer is which specific protein misfolds. In dementias, two proteins are mainly involved. The first one is the amyloid beta, which forms plaques that accumulate outside neurons between cells, which disrupts and hurts the cells. In the tau proteins, which is a structural type of protein, they tangle inside neurons, which collapses the cell's internal scaffolding. In Parkinsonisms, the responsible protein is alpha-synuclein. It coagulates inside neurons in structures called Lewy bodies, which is why it's called Lewy body dementia. These aggregates specifically form inside dope endocrine systems, in specifically the dopamine-producing neurons.
Dementias attack the hippocampus and cortex. These regions are responsible for memory formation and higher cognition. When the neurons die there, the brain loses its capacity to lay down new memories and reason clearly. The neurotransmitter primarily affected is acetylcholine. Parkinsonism attacks
The substantia nigra.
small region in the brain system. This region produces dopamine, which signals smooth, voluntary movement. When neurons die, the brain loses motor control. The result is tremor, rigidity, and slowed movement. This is our second major AP biology connection. Unit 4 covered cell signaling and synaptic transmission. When the sending neuron dies, the chemical message never reaches the receiving neuron. The downstream function, whether memory or movement, it breaks down.
We analyzed projected data from the Alzheimer's Association 2025 report. The line graph shows projected cases of Alzheimer's in Americans 65 and older, and in millions, from 2020 through 2060. We observed a roughly linear climb as it starts from 66 million cases in 2020 to 13.8 million in 2060. That's a 130% increase over about 40 years. Two factors primarily drive this. Primarily, the US population is aging, and age is the largest risk factor for both disease categories. Second, no curative therapy or cure currently exists. The treatments we have, which we'll discuss soon, slow disease but do not stop or reverse it. And without a breakthrough, this curve continues upward.
Until recently, both diseases could only be confirmed by autopsy. Modern imaging changed that. 3 techniques separate the 2 categories in living patients. That scan is used for Parkinsonism. It injects a radioactive tracer that binds dopamine transporters in
The substantia nigra.
A healthy scan shows 2 bright structures. A Parkinson's scan shows them reduced or absent. Amyloid, PET, is used for dementias. The tracer binds specifically to amyloid plaques and causes them to fluoresce on the scan. This confirms Alzheimer's pathology in a living patient. MRI is used for both. High-resolution scans reveal patterns of brain shrinkage called atrophy that match each disease's characteristic region.
Treatment approaches differ for each category. For dementias, cholinesterase inhibitors increase available acetylcholine, which produces a modest symptomatic benefit. The major breakthrough, though, is lecanemab, which is an FDA-approved drug in 2023. It is monoclonal antibody, which means it is an immune protein engineered to recognize and bind specifically to amyloid plaques. Once bound, the immune system clears the plaque through the T-body cells. In clinical trials, it slowed cognitive decline by approximately 27%. This is the first drug to address the underlying cause of Alzheimer's rather than just its symptoms. For Parkinsonism, the gold standard still remains levodopa, also called L-DOPA. It is a dopamine precursor that the brain converts into the dopamine that the patients have lost. In advanced cases, surgeons may implant deep brain stimulation electrodes that pulse the motor circuit and suppress tremors. Lecanemab is also our third major AP biology connection, as Unit 7 covered the immune system, and a monoclonal antibody is the principle of antigen recognition turned into a therapy, as our own immune system is trained to target a single specific protein shape.
To close, 3 principal findings from our investigation. First, dementias and Parkinsonisms diverge at the cellular level. Distinct proteins damage distinct neuron populations, which produces distinct clinical outcomes. Second, protein structure governs the seed pathway. The unifying molecular event across both categories is a protein folding into the wrong shape. That single fact connects Unit 1 of our course directly to a global public health crisis.
Third, targeted therapeutics are emerging. Lecanemab demonstrates that disease-modifying treatment, or otherwise known as treatment that addresses the cause rather than the symptoms, is now achievable. The next decade of neurodegenerative research will likely transform how these diseases are treated. Thank you.